Hello everyone, my name is Ankur and welcome to RJPT Simlab simulation. In this video, we will study about the effect of saline purgatives on frog's intestine. At the end of this video, we will be able to tell about the purgative effect by the use of hypertonic, hypotonic and isotonic solution on frog's intestine. So now let's start with the video. At first, let us see the introduction part. But let me tell you about one thing. What is a hypertonic solution? Hypertonic solution is any solution that contains a higher concentration of solute particles and a lower concentration of water particles in compared to a cell or any other solution, which means it is a solution which contains higher concentration of solute particles and a lower concentration of water molecules. Okay, and what is a hypotonic solution? A hypotonic solution is any solution that has a lower solute concentration and has a higher water concentration. It's just the opposite of hypertonic solution. Hypertonic solution contains higher particles, higher concentration of solid particles, and lower concentration of water. And hypotonic contains lower concentration of solid particles and higher concentration of water molecules. It's just the opposite. And now, what is an isotonic solution? Isotonic solution has the same solute concentration and osmotic pressure as any other solution. So, hypertonic and hypotonic solutions differs in concentration of solid particles and water molecule. An isotonic solution contains the same solute concentration, solid particles, and water molecules also. That's why it is known as isotonic. Now, let us know what is a laxative. Laxative is any drug used in the treatment of constipation to promote the evacuation of feces. Laxative produces their effect by several mechanisms. The four main types of laxatives include saline purgatives, fecal softeners, contact purgatives, and bulk laxatives. The statement says that it is any drug used in the treatment of constipation when the removal of fecal is not moved. It happens due to loss of water or when the water content is less in the body, when the body is dehydrated, so the movement of the fecal is not good. And for that treatment, laxatives are used. For the evacuation of feces, evacuation means removal. Laxative produces their effect by several mechanisms. It has very mechanisms. The main are four types: saline purgatives, fecal softeners, contact purgatives, and bulk laxatives. So now let us discuss these main four types of laxatives. At first, saline purgatives. These are salts containing highly charged ions that do not readily cross cell membrane and therefore remain inside the lumen or passageway of the bowel. By retaining water through osmotic forces, saline purgatives increases the volume of content of the bowel, stretching the colon and producing a normal stimulus for contraction of the muscle, which leads to defecation. Some of the commonly used salts are magnesium sulfate, Epsom salt, magnesium hydroxide, milk of magnesia, sodium sulfate, Glauber salt, and potassium sodium tartrate. The above paragraph says that salines are salts, basically salts which contains high amount of charged ions, highly charged ions, and that's why they do not cross the cell membranes and therefore remain inside the lumen. Lumen is the abdominal stomach portion. So due to that, they stay inside the lumen, and by retaining water through the osmotic pressure, they generate osmotic pressure and they retain water. They hold water and force osmotic pressure forces saline purgatives increases the volume of content of the bowel. So if water is retained inside the bowel or the stomach portion, the volume of water contents or the contents inside the bowel increases due to water, stretching the colon and produces a normal stimulus for contraction of the muscles. So due to the increase in the contents of the bowel, the colon stretches and produces a normal stimulus or normal act or force for the contraction of muscles. It contracts the muscles, which leads to defecation. Defecation is a biological cell process of the elimination of solid or waste materials known as feces from the digestive tract through anus. So defecation is the removal of the waste materials from the body through the anus. So on some of the commonly used saline purgative salts are magnesium sulfate, which is also known as Epsom salt, magnesium hydroxide, which is also known as milk of magnesia MOM, and sodium sulfate, which is also known as Glauber salt, and potassium sodium tartarate. So now let us know what is fecal softeners. Fecal softeners are not absorbed from the gastrointestinal tract and act to increase the bulk of the feces. Liquid paraffin can be used either as the oil itself or as a white emulsion. Other fecal softeners have a detergent action that increases the penetration of the stool by water. So we understand from the word fecal softeners, which means it softens the waste materials. They are not absorbed from the GI tract and act to increase the bulk of the feces. They are not absorbed from the GI tract, meaning they does not absorb water from the GI tract, but and increases the bulk volume of the feces. Liquid paraffins are the examples of fecal softeners. We can also say mineral oils can be used either as the oil itself or as a white emulsion. So in this case, liquid paraffin means oil phases are used for the softening of the waste materials fecal, and other fecal softeners have a detergent action that increases the penetration of stool by water. There are some other fecal softeners also which have a detergent action. So, which increases the penetration of the stool. It increases the penetration of the stool by water. So, in here, water is used, but most of the fecal softeners are oil phases. Now let us see what are contact purgatives. Contact purgatives act directly on the muscles of the intestine, stimulating the wave-like contraction peristalsis that results in defecation. This type of laxative includes cascara, senna, castor oil, and phenolphthalein. After regular use, their effect tends to lessen, so larger and more frequent doses are necessary until finally they cease to be effective. They are useful, however, when short term purging is required, example, before surgery or after an illness. So the above paragraph says that they act directly on the muscles of intestine. So contact purgative does not act on the waste materials or fecal, it acts on the muscles of the intestine. It stimulates a wave-like contraction, also known as peristalsis. Peristalsis is the movement of the materials inside the GI tract, or we can say intestines. So it generates the muscle contraction in the intestine and moves the fecal part and results in the defecation, removal of the waste material. So this type of laxative includes cascara, senna, and castor oil and phenolphthalein. These are the examples of contact purgatives. And after their regular use, their effect tends to lessen. So larger and more frequent doses are necessary until they cease to be effective. So if we use these drugs regularly on regular basis, so due to that, it will lessen the effect of these drugs. So larger amount doses are needed when they are used regularly to seize the therapeutic effect. So they are useful, however, when short-term purging is required. So these are used when short-term purging is required. Short-term effect is required. As we can say, for example, before surgery. So before surgery, these drugs are given so that instant or short-term action is seen of these purgatives, or after an illness because the body is weak. So the patient needs contact purgatives for that. Now let's see bulk laxatives. They act by increasing the size of the feces, in part because their capacity to attract water. This group includes methylcellulose and carboxymethylcellulose, the gums agar and tragacanth, psyllium, seed, and dietary fibers. So the bulk laxatives are they increase the size of the fecus. It acts on the fecus and increases the size, and in parts because of their water attracting capacity. They attracts water, increases the size of the fecus, and due to this, and this group includes examples, as we can say examples of bulk laxatives are methylcellulose and carboxymethylcellulose. They are cellulose that they absorb water and increases the size of the fecus. Some of other examples are gum agar and tragacanth, psyllium, platango seeds, and dietary fibers. As you know, dietary fibers also absorb a lot amount of water. So let us learn about this through an example. Through an experiment, we will perform an experiment on frog's intestine. So the experimental requirement is, at first we need the experimental animal frog. Then the second we need The requirements are frog's board, dissecting instruments, pithing needle, needle with thread, syringe with needle. Then the drugs and solution that we require for this experiment are magnesium sulfate of 27%, and it is an hypertonic solution. Then second, saline solution. Saline solution is used of 0.5%, and it is an hypotonic solution. And frog's ringer. Frog's ringer is an isotonic solution. We have already learned what is hypertonic, hypotonic, and isotonic solution. Now let us see the procedure for this experiment. The first step is pith a frog, which means we have to pick a frog and we have to place it on the dissecting plate or dissecting board. Then expose the abdominal cavity. We have to face the frog upwards and expose the abdominal cavity, and we have to cut the abdominal skin portion for the exposure of the abdominal cavity. Cavity is the hole. So trace the small intestine and make three compartments by tying threads at equal distance. We have to trace the small intestine. We have to place it out with the help of forceps, and then we have to make three compartments of this intestine, small intestine, and tie threads at equal distances. We have to tie the threads in the intestine at equal distances. Then secure the threads tightly so that no fluid can seep through from one compartment to another. So we have to tie the threads so that no fluid passage can be allowed, and the water is kept in each passages, in each compartments. So in the first compartment, we inject 0.2 ml of hypotonic solution. The hypotonic solution which we are using is saline solution. And then in the second compartment, we inject 0.2 ml of magnesium sulfate. We inject magnesium sulfate, which is the hypertonic solution. And then in the third compartment, we inject 0.2 ml of frog's ringer, which is our isotonic solution. And then wait for 20 minutes and record the observation. This is the observation table of this experiment. We will discuss it later while performing the experiment. So now, let us perform the experiment. This is the RJPT Simlab simulatory laboratory. On the left side of the toolbar, we can see our animal frog, which we will dissect and open. On the right side of the toolbar, we will see the dissection tray and syringes. The dissection tray is needed for the placement of the frog, and with the help of the syringe, we will inject our purgative drugs. And on the lower part, we will see the instruction part and the start and stop button. So now let us start the experiment. At the first, we have to put the frog on the dissection table. So as we can see here, we will place the frog over here on the dissection table. Here we have put the frog, so it is stable. And we will place it on the dissection table very carefully, and the abdominal portion is facing upwards. Now, from the next step, we now dissect and pin the frog. So with the help of scissors and forceps, we will cut open the abdominal portion of the frog so that the internal organs are easily see-through. As we can see, with the help of the scissors, we are cutting the first layer of the skin of the frog, and we will elevate it with the help of forceps. On both the sides, we cut and open so that the internal organs are easily visible to us. So now the skin is elevated of the frog, and now after the elevation of the skin, as we can see, the internal organs are easily see-through, and now we will pin the elevated skin so that it does not hinder or move while performing the experiment. With the help of these 4 pins, we will elevate the skin and so that it stays stationary in position. So now let us move on to the next step. Now we will stretch the small intestine of the frog by clicking the start button. As we can see here, we will stretch the small intestine with the help of forceps. We have stretched the small intestine portion. Now we will move on to our next step. But we have to carefully do this step because the internal organs and the small intestine is a very sensitive organ, so it does not break or damage. Now we will move on to our next step. We have to tie the intestine in 3 compartments. So with the help of forceps and scissor, we will tie threads on the small intestine and make three compartments. As we can see, with the help of forceps, we are tying knots on the small intestine. We have tied one at one point, then we are tying at the another point, so that three compartments are made and no passage of the fluids inside the compartments are seen. We have to tie that thread very tightly so that no passage of the fluid is seen. As we can see, we have tied the thread in the small intestine. Now we will move on to our next step. Now we will add drugs in each compartment, in each three compartment. As we know, we will insert the first compartment with 0.2 ml of 0.5% Na sodium chloride saline solution, which is our hypotonic solution. And on the second compartment, we will inject the 0.2 ml of 27% magnesium sulfate, which is our hypertonic solution. And on the third compartment, we will inject 0.2 ml of Ringer solution, which is our isotonic solution. So now with the help of the three different syringes, we will inject the drugs in each compartment and we will throw these syringes after their use so that it stays sterile and no chances of microbes are seen. So now we've injected all the three drugs in each compartment and then we will move on to our next step. Now we will observe it. We will inspect after 20 minutes. So now let's see the effect of these drugs. As we can see here, we have made three compartments and we are observing that at the first compartment has been shown and the second compartment has shown its effect. It can be seen it is swollen, and the third compartment has shown no change because of the isotonic solution. The isotonic solution has shown no change in the lumen. In the second compartment, magnesium sulfate, the hypertonic solution, it has shrunken. As we can see, the lumen portion is enlarged and the particles are shrunken. And then in the first compartment where we have used the sodium chloride saline solution, it has shown its effect that it has swollen the lumen. So now we will see the observation table. The first compartment has shown the first compartment over in which we have injected the 0.2 ml of 0.5% sodium chloride saline solution, which is our hypotonic solution. The observation shows that it has swollen the lumen part. Then in the inference says the hypotonic solution caused swelling of the tissue as fluid moved into circulation, which means that it has, due to the hypotonic effect, it has caused the swelling of the tissue by moving the water fluid inside the lumen. The second compartment in which we have injected the 0.2 ml of 27% magnesium sulfate, which is our hypertonic solution, has shown shrunken, which means the hypertonic solution moves the fluid from circulation to the GI lumen, thereby shrinking of the tissue. So by this, the shrinking of the lumen part has been observed. And in the third compartment, the 0.2 ml of Frog's Ringer solution, which is the isotonic solution, has shown no change because the isotonic solution did not show any fluid movement across the intestinal membrane. Hence, no change in the tissue. So, with the help of this observation table, we have learned about the effect of the hypertonic, hypotonic, and isotonic solution of the purgative effect on the frog's intestine. So, now we have completed our experiment. When we click the effect of saline purgative, the title of this experiment, we will see this page. This page shows the written portion of this experiment, the introduction part, the procedural part, and the observation table part. We can go through this before performing the experiment so that we have a brief idea about the experiment. And on the download user manual, when we click here, we will see the written portion of the experiment in a PDF format. This is for those students, those who do not have our RJPT Simlab subscription, so that they can at least have a go through and a brief knowledge about the experiment. So now we have completed our experiment over here. I hope that you have learned a lot about in this experiment, and thank you for watching this video. And and I hope that this video was very informative for you. Thank you guys for watching this video, and for more such learning content, subscribe to our YouTube channel RJPT Informatics and follow us on our LinkedIn profile, on Instagram as well as in Facebook. We upload and update our such learning content over these platforms. Thank you.